Thank you, Brianna. Thanks. It's a great pleasure to talk at the Targeted Protein Degradation seminar and symposium. I don't normally care too much about proteins in my career. It's always been on RNA, unless they've been making or binding RNAs or carbohydrates. So it's great to talk to you guys. We're in a strange area of RNA. I think RNA has been in the background, but in the past 2 years, it's certainly been in the foreground for a great many reasons. Not only has an RNA changed world history for us, but it's also changed in a negative way, but it's also changed it in a positive way. We're now in the era where messenger RNA is a medicine, which is, I mean, the Moderna, BioNTech, Pfizer vaccines are being delivered to people in nanoparticles, lipid nanoparticles. I don't know. I'm getting goosebumps talking about this right now. I mean, there's an era where you guys that haven't worked with RNA think that if you touch a tube that has an RNA in it without a glove on, the RNA is going to disappear. And yet we're injecting that into a human and it's providing life-saving medication. I don't know. It's sort of breathtaking. So today I'm going to tell you about work where, the medicine is an RNA, but we're trying to develop compounds that bind to RNA structure and affect its function. I want to start by thanking the people in the lab. Like many of you, I have a young family. I have 2 young children with my wife, and it's certainly been a challenge during COVID to maintain some level of sanity through, the craziness that is trying to figure out how to give schooling and all this stuff. And I think one of the calming things in all of this has been the lab, right? The lab has had craziness. However, right, being able to go in lab and do science and address problems has really, for no other time in my life, has really showed me the importance of working with young people, working with people, not young and old, anybody that's interested in science has been really important. So I'm gonna tell you today about work that was done by Matt Costellas, who's now a scientist at Dicerna, Hafiz Hanif, who's a scientist at Skyhawk, Raphael Benhamu, who's a professor at the University of Jerusalem, and Ali Angebello, who's a scientist at Expansion Therapeutics. So this is my current group. Actually, it's not so current, it's about 6 months old. I want to thank the people that funded this work, including the US taxpayer. I don't think the US taxpayer gets enough credit for the work that they've been doing in funding basic science, funding the basic science of T7 RNA polymerase that makes RNA. Now we're making medicines by using this enzyme, and that was done by Bill Studier, Brookhaven National Laboratories in the 70s, and then Aki Uhlenbeck and others have worked to make that into making RNA. People like Lynn McQuat, Melissa Moore working on studying RNA quality control mechanisms and how to shield messenger RNA, how to know that messenger RNAs get exposed to them and how to shield them from them is part of how we have RNA as a medicine now. It's really amazing. All right, so I've broken this into 4 sections. This is the introductory section. And many of you don't think about RNA. So it's just sort of an overview that I wrote in JAX as a perspective on why I think targeting RNA is important with small molecules. And so I do realize I'm in the lion's den, so to speak here, of people that think about protein. But I want to, about 1 to 2% of our human genome encodes for protein, and yet 80% encodes for RNA. As many of you are intimately aware, you've devoted your scientific lives to this. Even targeting a protein doesn't mean it's a panacea. Even though 1 to 2% of our human genome encodes for protein, only about 15% of protein families have been drugged with a small molecule. And so the term for the rest of those proteins are the quote unquote undruggables by people like Greg Verdine. And so a lot of work has been done on figuring out how to drug this 15% more, the proteome more broadly. And the impacts of that to patients have become very apparent even in the past few years. So Showkat and colleagues have tried to study how to covalently target things like mutant KRAS. And now, because of that basic work that an academic institution has done, there are Mirati and Amgen have clinical compounds that are targeting mutant KRAS and really changing the course of medicine for people that have cancers that wouldn't be able to have hope a few years ago. I mean, it's really profound. Craig Cruz, Ray Deshays, Jay Bradner, Nat Gray, Carolyn Bertozzi, people working aggressively in the area of protein degradation broadly. A lot of those molecules are being brought to the clinic. There was a review in Nature Reviews Drug Discovery that I just saw last week that was published, and there's like almost 24 different compounds that are in clinical patients. Man, if nowadays we don't realize the value of basic science to study molecular recognition and do things and get that into a patient, we're never going to get that message. So that's really important work. What my lab has been working on, and from my entire academic career, I just got an email right before this talk from the guy I did undergraduate research with, Jeff Davis. And as an undergrad, I was studying RNA folding, isoguanosine bases in chloroform. And then, almost 20 years ago. And from that, I had an interest. Jeff really catalyzed me into working and doing science. He's really a wonderful person, and we share a love for baseball because today is opening day and I'm talking to Boston, and I'm from Baltimore, and the Orioles were playing the Red Sox, but that game has been postponed because of weather. So nevertheless, I've had a long-term interest in how RNA folds, and I got my PhD with Doug Turner, who's sort of the godfather of RNA folding. And yet when we look about RNA, we don't think it folds, and about 0% of it has been drugged with a small molecule, in part because the selectivity challenges to try to drug structured RNA has been so profound that it's just really hard. That being said, there has been a wide variety of antisense oligonucleotide-based approaches that have targeted RNA. Spinraza, an RNA splicing medicine that has been treating patients with spinal muscular atrophy. Allele is right down the street from you guys. That is targeting a bunch of liver-associated diseases by using siRNAs. And so those have been game-changing modalities, but yet there's been very limited, in fact, very little activity in making small molecules that target RNA. And we wanted to fill this space very aggressively for the lab over the past 16 years because we thought RNA was a folded molecule, just like these proteins. The question is, how could you identify RNAs that are folded, and how could you stick a compound into those folded sites to affect the function to study it? Now, the reason that we wanted to study this is not just for basic science, but we know that both coding and non-coding RNAs literally play a role in nearly all diseases. And so this is about a year and a half old. And so you can see if we just go on PubMed and we look at the diseases that are caused with RNA, these are really important ones. It's infectious diseases. People are targeting SARS-CoV-2, my lab and others, because it has an RNA genome: diabetes, cancer, heart disease, Alzheimer's, fibrosis, dementia, ALS, myotonic dystrophy, are all can be traced to a structured RNA that causes the disease, that if you could target it specifically with a small molecule, you could affect its biology. And so that's what we aim to do broadly in our program for a decade and a half. So I'll tell you about the way that we think about targeting RNA broadly. And what I'm going to show you here are chemical probes. We're not going to, it's quite a barrier to make a medicine, as many of you know that are involved in doing medicinal chemistry, than it is studying chemical probes and the effects of molecular recognition. Okay, so the way that I view RNA is a line of nucleotides that are shown here to the left, and then it will collapse into a folded structure. So what we've been able to do is, because of working with Doug Turner, we thought that we could computationally study druggability of the transcriptome from RNA sequence. And so I'm just broadly going to go into this, and then I'm going to tell you some examples for how we've been able to apply this technology. And so if we look at an RNA, RNAs generally look like this. There are regions within an RNA that have a high probability of a structure, and there are regions within an RNA that have a low probability of structure. And if you're looking at a region of an RNA that has a low probability of structure, you'd want to target that with a oligonucleotide that recognizes the base pairs within that structure. If, however, you want to target an RNA that at a site that is highly structured, then you want to target it with a small molecule. And so we've developed technologies that allow us to look at these regions of structure and ask a question and say, is this a druggable pocket? And much like druggable and undruggable was positioned early on in terms of, do you have a molecule that can bind to it? We study druggability or ligandability by if we have a small molecule that binds to a particular RNA structure in a database that we call Infauna. And so we can have monomeric molecules that bind to targets, or we can make compounds that bind to two different RNA folds that are in close proximity. And so one of the things that we also have developed over the course of more than a decade are technologies to understand what are the molecular recognition events that are occurring between small molecules and RNA in cells. And so we developed an approach that we call chemical cross-linking and isolation by pull-down, in homage to Bob Darnell, who developed CLIP, which is a way to cross-link proteins for binding to RNA targets. And basically in a cell or in vitro, small molecules will go in, recognize the RNAs they bind, and they'll undergo a proximity-based cross-linking event. We can purify those cross-links and then identify what are the RNAs that are bound and the sites of binding. Now, in the cases I'm going to talk to you today, they're all going to elicit a physiological effect by binding. But there are many cases, as we've been able to show, is that binding of a small molecule to an RNA target by itself, in fact, most of those cases are not biologically functioning. That is, a molecule can sit there on a messenger RNA open reading frame. If it does not have sufficient thermodynamic stability that is provided to the RNA, a ribosome will read through it, kick off the protein, and there'll be no effect on function. For those cases, we sought to develop a generalized technology that could facilitate those compounds for being bioactive. And what we did is we basically can make a secondary molecule that'll bind to and sense ribonucleases in cells and animals that'll trigger the catalytic and substoichiometric degradation of RNA targets by this ribonuclease targeting chimera approach. But what I did in this talk, because I think many of you don't think about RNA. I think about it all the time. It's all I've ever wanted to do was study RNA, ever, from an undergrad to a graduate student to now. I think it's simply amazing that a molecule, a biopolymer built of 4 bases, is able to adapt functional folds that can have both coding and non-coding functions that can do things like silence chromosomes, can repress protein translation on a broad scale, and even more profoundly, nucleotide repeating transcripts can cause genetically defined diseases like Huntington's disease, which, even though a clinical trial was unmasked this week by Wave and Biogen Ionis, and it didn't have efficacy for the patients, I do think that if we can unlock ways to target these aberrant RNA structures, we could impact disease broadly. Now, what I just showed on that last slide was targeted degradation by ribonuclease recruitment. There are 3 ways, and this is how I've broken down the rest of the talk, that we have addressed and applied ways to affect targeted degradation of RNA. Now, I've been paying close attention to the talks that you guys that have been given in this seminar, like many people have. This is really great, by the way. And so I'm going to tell you 3 different flavors by which we've been affecting the targeted degradation of RNA in cells. So the first way is that there are many diseases where a toxic RNA is an intronic RNA. And you can ask yourself a question, go, hey, Matt, how is an intronic RNA causing a disease? Intron, as Phil Sharp down the street, you guys are in the mecca of science, like, does it ever end? So at MIT showed is that introns get spliced out of messages and they get degraded by the RNA exosome in the nuclear exosome. But many diseases are caused by intronic RNA repeats that are harbored as retained introns. That means the rest of the RNA is properly spliced, except that version that has that intron that has a toxic repeat. And Maury Swanson at the University of Florida discovered that these introns are harbored as retained introns. And what we've shown recently is that small molecules can bind to these retained introns and force them to be processed naturally, where they're liberated as a normally retained intron and they're subjected to degradation. So this is a way that a small molecule can bind to a toxic RNA and facilitate native quality control degradation. The middle is we've been able to degrade targets for a while by appending natural products onto RNA binding compounds that facilitate oxidative cleavage. So bleomycin and its derivatives where they don't cleave DNA. And by doing that, you can basically undergo a proximity-based degradation event where you'll undergo a strand cleavage of an RNA. You'll liberate an RNA from its poly A tail, and it'll be degraded by this bleomycin or direct degradation. And then the end of this talk, I'm going to talk about induced proximity and activation of a ribonuclease way to degrade, where we have small molecules that bind to an RNA target. Now, small molecules I'll get to later. And then one molecule will recognize an RNA target, the other will recognize a ribonuclease. And in the case of RNA cell, which is the only ribonuclease that we published to date, it's an inactive monomer that the compound induces the proximity of this ribonuclease and dimerizes it. So it's like Stuart Schreiber and Craig together with Ray. It basically superglues the ribonuclease together to activate it and induces its proximity to facilitate targeted degradation. And we've demonstrated all of these approaches work in cells and animals. But here are some advantages and disadvantages for this. So first is that this nuclear RNA exosome mediated degradation, you can get very low molecular weight drug-like rule of 5 obedient small molecules that affect this pathway. We have a lot more work that's coming out in an accelerated way for this across different disease settings. Because this is taped, I'm just talking about the published stuff, but I'm happy, you'll see more about this early on. These compounds can be very specific. It's not a lot of RNAs that have retained introns, it's very few, and if you facilitate its degradation, it can be very specific for that. The disadvantages of this approach is it's only applicable to a number of targets, and it generally only targets nuclear RNAs, because these RNAs are subjected to a nucleus, and you have to facilitate its degradation by having the spliceosome recognize an RNA and liberate that retained intron. The oxidative cleavage approach allows us to target both nuclear and cytoplasmic RNAs, and it can be quite potent, where we can degrade 90% of a given RNA target. The disadvantage is that it's not rule of 5 like, and that the degrader moiety, given that it's bleomycin or its derivatives, can have a broad perception of having significant limitations. Some of that is real and some of that is not necessarily real, and we can talk about that more. And then last but not least, this ribonuclease targeting chimera approach, we've demonstrated that this works substoichiometric. Actually, I believe all of these approaches work substoichiometrically, but we've most demonstrated it with the RiboTac approach. I believe this advantage could also be a disadvantage. So I was trying to be an optimist this morning, given that it's the opening day of baseball and my team is the Baltimore Orioles. I have to be an optimist today of all days, is that I think this has a great potential for optimization. That is, there are many nucleases one could be able to recruit and many programmability that this can have. This is also probably the 2 degraders to the right. It can be general. There's a lot of targets that you can affect this against. The disadvantages are it's not rule of 5 like, that the current RNA-L degrader prefers greatly cytoplasmic RNAs. And the first gen molecules, now I think we can optimize this by dosing and half-lives of RNA. They degrade about 40 to 70% of the target, right? And this is early, right? One of the luxuries, I think, I want to be sensitive to you guys. One of the major advantages, I don't want to call it a luxury, an advantage of working in the targeted protein degradation space is there are so many ligands that target a protein. So you could take a kinase inhibitor and turn it into a protein degrader, and that can be majorly advantageous. There's not a lot of bioactive ligands that target an RNA that you could immediately turn it around to a degrader. And so the challenge is the degrader medicinal chemistry and the binder medicinal chemistry. There's not an easy way to short circuit that. All right, so I'm going to tell you about how a monomeric degrader can trigger destruction of a repeat expansion, okay? So we applied this approach in the first 2 sessions to RNA repeat expansion. So these are 40 diseases that have no known cure. These are like really important diseases. So this is the repeat expansion that causes Huntington's disease, which was the 2nd disease gene after the cystic fibrosis gene that was associated with a chromosome. Dave Hausman, Jill Bates, Nancy Wexler worked on this. It's a great history lesson for the students about human genetics. Hausman and his team also discovered myotonic dystrophy type 1, which is the most common cause of adult onset muscular dystrophy. And most recently, the most common cause of ALS was associated with a toxic repeat expansion that caused G4C2 mediated ALS. And so these repeat expansions can cause disease in very different ways. They can bind to a protein via RNA gain of function. They can be translated without a canonical start codon, and they can be misspliced. I'm going to focus on RNA gain of function and this missplicing of a retained intron in this talk. So these repeat expansion, I'm going to talk about a CUG repeat expansion that causes myotonic dystrophy type 1, which is the most common form of adult onset muscular dystrophy, and Fuchs' corneal dystrophy, which is a common front of the eye disease. These two diseases that are very different are both caused by a CUG repeat expansion that binds to proteins. In the case of DM1, it's a CUG repeat expansion that's in a 3' UTR. In the case of FECD, it's an intronic repeat expansion of CUG that's in intron 3 of this TCF4 messenger RNA. Now, in both of these diseases, they're caused because muscleblind, which is a broad RNA splicing regulator, is bound to and sequestered by this CUG repeat expansion. What happens is muscleblind, what it normally does is it recognizes pre-messenger RNAs, and it binds to RNA structures that look very different than the repeat expansion structure in pre-messages and triggers the spliceosome to exclude exons. In the case of the muscle-specific chloride ion channel, muscleblind will bind to two structures that are 5' of exon 6B and 7A. In the splice form, it'll exclude those exons, and you get this form of the pre-messenger RNA, which forms functioning chloride ion channel, and you get normal muscle function. If, however, the muscleblind does not bind to the pre-messenger RNA, but stuck on the repeat expansion, you can include exon 6B and 7A. And then this messenger RNA has a premature termination codon, which, as we were told by Lynn McQuat and Melissa Moore, who've most studied this pathway, Melissa Moore being the current CSO of Moderna, Lynn McQuat, I hasten to say winning, but earning the Wolf Prize last week for studying this decay pathway, triggers this messenger RNA for being subjected to nonsense-mediated decay, and it's gone. If, however, we take a small molecule that can bind to this target, it'll force muscleblind to bind to the natural pre-messenger RNA, and that'll facilitate RNA splicing that we want to get this fully functioning channel. Okay, so I'm not going to go into the design, but suffice it to say, we designed this very undrug-like ligand that targets CUG repeats. When we throw this compound into myotonic dystrophy type 1 patient-derived cell lines and Fuchs' corneal dystrophy patient-derived cells, the drug acts similarly. It's the same repeat with two different diseases, and the drug will selectively improve RNA splicing defects in both DM1 and FECD. Notice that FECD is a bit more potent than DM1. We might come back to why this is the case, or we're going to come back to why this could be the case later on. One of the advantages here is if you think about, I'll talk about this later, but ASOs that target these genes do not target the repeat themselves or oligonucleotides. They target the gene that harbors the repeat because it's so hard to get an antisense oligonucleotide to bind to these repeating targets with high potency, and it's very hard to get it to be selective, as I'll talk about later. Now, as I'm going to reiterate here that in FECD gene, in healthy cells, this intron 3 and this TCF4 is liberated. You never get this built up intron. But when you have a large repeat expansion in FECD cells, as we've shown, it binds to muscleblind, and muscleblind binding to this target does not allow the spliceosome to recognize this intron as an intron, and it gets harbored as a retained intron. And in this paper, and this was, again, an observation initially by Mari Swanson, in this paper, we can transfect in muscleblind, we can siRNA ablate muscleblind, and it's very clear that muscleblind binding itself controls how much intron is retained. Now, what I just showed you on the previous slide is that when we add the drug, we improve splicing, which means that we're reequilibrating muscleblind to be off these repeats and onto the native target that it affects the splicing of. What is the consequence of the gene? Of this intron. And so what we can see in DM1 patient-derived cells, there's no effect on the abundance of DMPK message. In Fuchs' corneal dystrophy, there's a diminishment in the level of this retained intron. And in fact, that CUG repeat expansion that is intronic is liberated and subjected to decay. That we know that at 20 micromolar, we're getting back to the wild type levels where literally the small molecules binding to this RNA, facilitating its liberation. It's no longer retained intron, and it's being degraded by the nuclear RNA exosome. So we've been able to show this by, this is paper one, we have others in this to really nail this pathway, that we know that what's happening, again, is this ligand's liberating muscleblind, then you're getting splicing and debranching, you're liberating this intron, it's no longer a retained intron, it's a liberated intron, and the RNA exosome is facilitating its targeted degradation. So it's a monomer can facilitate degradation as a binder. You would call this monomer, no, probably not, because that would be a molecular glue, but this is a monomer that affects RNA processing to facilitate its targeted degradation. And this is general. We've observed this not just in DM1, not in Fuchs corneal dystrophy, but not DM1. We've also observed this with type 2 myotonic dystrophy. So myotonic dystrophy type 2 is caused by a CCUG repeat that's in intron 1 of the ZNF4 pre-messenger RNA. And when we treat drugs in DM2, you can facilitate the targeted degradation of this repeat. What is interesting is that muscleblind itself has CCUG repeats. That's not toxic. But if you take an ASO, the ASO will target those CCUG repeats and degrade this, the disease-causing gene that causes myotonic dystrophy type 2, but it also degrades muscleblind. So we believe that these small molecules that can facilitate targeted degradation can be very important for many of these rare diseases. And we've been able to show that all of these diseases that are caused by an intronic RNA, you can facilitate its targeted degradation by one of these binding molecules. And we've been able to get this to work not only in cells, but also in animal models for these diseases. Okay, so now I'm going to switch gears here and I'm going to talk about direct targeted degradation of a repeat expansion, where we're using this oxidative cleavage by bleomycin. And again, so what I'm going to talk about here is not monomeric binders. We didn't know this pathway existed when we did this, but I'm going to talk about compounds that target 2 sites on this RNA target simultaneously. Okay, so it's dimeric molecules where we're recognizing the structure that the repeat expansion is forming with compounds that target 2 of these sites simultaneously. Now, these are not drug-like compounds. I've tried to be clear here and say they're not traditional small molecules. You guys live in this space as much as I do, but I think the only other way to degrade an RNA would be an siRNA, and these molecules certainly have more drug-like, I shouldn't say drug-like properties, they have more the ability to do medicinal chemistry on them than an ASO, I would say. Now that they're not developed like an ASO, they haven't reached patients, but one hopes that they could, and there might be some synergy between diseases that an ASO can target and maybe these would, I don't know, but for our case here, I'm describing these as chemical probes to study molecular recognition in an animal. Okay, so that dimer is bioactive at 100 nanomolar compound concentration. It'll improve the splicing defect of muscleblind. I'll get to questions at the end. Maybe, do I want to answer questions? I'll get to questions at the end. I know some are being added. So this dimer at 100 nanomolar compound concentration improves about 60% of the RNA splicing defect. As I mentioned earlier, and I reiterate here, is we've developed ChemClip as a way to study direct target engagement in patient-derived cells and animals, and you can read it out by pull down, RNA-seq, et cetera. And so this is DMPK message. This is the messenger RNA that harbors this large repeat expansion in the 3' UTR. And when we take a ChemClip probe, we can enrich and pull down DMPK message. When we add competitor, this non-reactive, non-cross-linking molecule, we can deplete the pull down. We also have been able to map the binding sites for ligands on RNA targets by using this approach. Frankly, it's quite easy to do that by next-gen-seq if you have a non-repeating RNA target. But one of the focuses that I had in my lab, because my niece has a rare disease, has been working on these rare disease repeat expansions. And so we had to develop, and it's hard to read through a fully repeat expansion by next generation sequencing. And so we had to play a trick here to profile ligand binding site into these targets, where basically we pull down, this is the anatomy of DMPK message. And then we basically used antisense oligonucleotides in the pull down fraction and can site specifically make a nuclease to chop this RNA target into bits. And then we could see what fraction of these RNAs that we capture based on digesting it into individual regions. And we know that the only region that we pull down is the one that contains the CUG repeat expansion, validating that our ligand is recognizing that repeat expansion in a cell. And it's an allele selective molecular recognition event, and I'll get to that later. The other thing that we did is we wanted to see if we could make a direct degrader, and some work by Sid Hecht has suggested that bleomycin, one of its targets could be RNA. So, we thought, well, we didn't have the ribotac fully baked at that point. We thought, oh, could we use this bleomycin cleavage as a way to profile binding sites? And we did that in cancer, where you can make these compounds that have this bleomycin cleavage. It'll bind the target, it'll cleave it, and you can reverse amplify the site of cleavage. We can use this to map the binding sites. And the other advantage is that it allows us to recognize and read out, as you guys are well aware of this, but in the RNA space, we didn't have these tools until they were developed. Our labs played a major hand in that, where we could read out what are the RNAs that are bound in a cell in an animal by using cleavage as a readout. Now, this is really important because cleavage it's very hard to purify cross-linked material from animals. And so what we really wanted to do is utilize this cleaving approach to read out molecular recognition events in a mouse model. And so this is work by Ali Angebello and Suzanne Zuchek. I introduced them in the beginning. And so basically we made these monster compounds which have these CUG ligands that are attached to bleomycin. Now, the way that we attach the bleomycin is we made this urea conjugate to the binder. And it turns out the positive charge of this compound on bleomycin allows DNA binding. And when you block it with this urea, you ablate that dramatically. That's illustrated here with this gamma H2AX staining. You can basically see that you don't generally get this gamma H2AX staining with these compounds that are directed to bind RNA. And the other thing we've been able to do, Ali has removed the carbohydrate portion of this with the D-glycobleomycin in a collaboration with Dale Boger, and that molecule has an even more ablated effect on targeting DNA. We don't get any DNA degradation. Okay, so one thing we did is we took these degraders and delivered it to patient-derived cells, and we first asked the question, said, if we use a oligonucleotide that recognizes a CUG repeat sequence, what is the consequence of all the genes, all the messenger RNAs that can change short and non-toxic repeats of CUG in a cell? And this is a patient-derived fibroblast, but we get the same thing with a myotube. And you can see all of these RNA targets, and this is the CUG repeat number that it has, are diminished when you have an antisense oligonucleotide that targets the CUG sequence. If, however, we have a structure binding ligand that facilitates targeted degradation, we only get ablation of the mutant allele of DMPK message. And I can tell you these two compounds are dosed at the same amount of molecule that improves, I believe, 50% of the RNA splicing defect. And so you actually, this is, I think, a fair comparison. So, but nevertheless, you can get this allele selective molecular recognition for targeted degradation in a patient-derived cell line. We then worked in a DM1 mouse model to see what was the effect of these compounds in a mouse model, knowing that what we wanted to do is see if we could degrade this target, how much we needed to degrade, and could we have the ability to have these compounds facilitate an improvement in myotonia, et cetera, within the mouse model. But we really wanted to study molecular recognition events in an animal. We had ChemClip at our disposal to study what are the RNA targets that are bound and not bound in a cell, and we know that we can get allele selective recognition, but we didn't know what would be the consequence of delivering these in a mouse and what were the targets that we would hit. Okay, and so first things first is we can degrade about 40% of this DMPK message in this mouse. At these doses here, this is a 10 mg per kg IP every other day for 2 weeks. That we can ablate about 40% of the messenger RNA with this compound that we call cougamycin in both the TA and the gastroc muscle in these mouse. And that improves about 50% of the RNA splicing defect. That improvement of 50% of the RNA splicing defect is correlated with an improvement in the myotonia of these mice and also correlated with the increased expression of that muscle-specific chloride ion channel. And so what's stained here in green is the expression in the mouse of the muscle-specific chloride ion channel. Here's a DM1 mouse. You can see that goes away in when it's treated with vehicle. However, when it's treated with this degrader, you can see that it's massively upregulated because we've corrected the RNA splicing product that's formed from the muscle-specific chloride ion channel. We've gotten rid of this premature termination codon to allow the muscle specific chloride ion channel to be broadly expressed in a DM1 mouse and improve the myotonia. The next thing we did in collaboration with Eric Wang is we pulled the mouse muscle and we wanted to see how selectively could these molecules that bind this target improve an RNA-mediated defect. So each one of these dots is a splicing event that's dysregulated in a DM1 mouse that is HSALR versus a wild type mouse. And this is when we deliver it with our compound. You can see broad compression in these RNA-seq measurements. About 97% of the pre-messenger RNA splicing defects are improved in this DM1 mouse model. And the other thing that we were able to do is run gene expression profiling. And so here's the gene expression profiling. What I showed previously was splicing. This is gene expression profiling. And you can see these genes are broadly compressed by about 20,000 of them with the drug, suggesting that we're improving splicing and gene expression profiling. Last but not least is we studied the ability of these molecules to affect gene expression profiling in a background mouse, and we didn't see any effect in gene expression profiling in these background mice. These are all of the short CUG repeat containing genes that are expressed in the mouse. This doesn't have the mutant allele. Because it's a wild type mouse, the mutant allele would be degraded to about here off this diagonal. And you can see there's no effect on these shorter non-toxic repeats of CUG. This is an allele selective molecular recognition event with a structure binding ligand. Okay, so I'm going to end in the last 10 minutes by talking about targeted RNA degradation via ribonuclease recruitment, this ribonuclease targeting chimera, which is probably what most of you guys thought I was only going to talk about. And so this is work, a PNAS paper. We've worked on we've tried to recruit these nucleases for probably a decade, and we have various other nucleases that we can recruit. This is the most fully baked one, and the one that we were, you'll understand why we were interested in this. Okay, and so we wanted to apply this to not a human genetic disease, but we wanted to apply it to cancer. And so metastasis obviously is a cause of death in cancer patient. And long story short, many RNAs are upregulated in and causative of metastatic cancer. The target we went after here is microRNA 21, and microRNA 21 is a non-coding microRNA. So Gary Ruvkin and Victor Ambros identified these from fruit flies, and they're broad dimmer switches that are expressed in the human transcriptome. And so microRNA 21 is upregulated and causes metastasis in triple negative breast cancer. MicroRNAs are made as a primary transcript that are processed in the nucleus by the nuclear nuclease called DROSHA at this site to liberate a precursor microRNA that gets translocated to the cytoplasm, where it's degraded by the cytoplasmic or processed by the cytoplasmic ribonuclease called dicer to liberate a mature microRNA. Basically, that mature microRNA recognizes the 3' untranslated region of messenger RNAs by which it has base pairing complementary to, and it represses or turns down how much protein is made from that messenger RNA. And so in that way, this will silence P10, PDCD4, and Spridey, and that'll cause invasion and metastasis in these non-coding RNAs. And so one way to inhibit them, which we've shown, is you can have compounds that bind to the nuclease processing sites in either the primary prime microRNA or the precursor microRNA. And so we looked at, by using our software, I'm not going to go into this, what are the RNA targets that we could bind with a small molecule, and what are the off targets in the human transcriptome computationally. And so long story short, if we want to target the dicer site, there are 20 other microRNA precursors that contain this motif. But if we look at the adjacent site, there are 39. There's only one target that has both of these sites in the human transcriptome. So we made a dimer that binds both of these sites. And we went into Inforna and we could identify 2 fragments. They're large fragments. I'll show you the structure later. They're not very drug-like, but it's a chemical probe to study structure binding ligands can bind the RNA target. And here's a model of the molecule sitting on the 3-dimensional shape of this RNA target. You can see, even though I've rendered these things in 2D structures, they have a 3-dimensional shape. I don't want you to think they're not 3-dimensional. They are. Okay, so if we take the monomer and the dimer and we test it in triple negative breast cancer cells and we measure what's the effect on microRNA 21, you can see that in orange, this is the monomer. At 10 micromolar, it inhibits about 50% of microRNA 21 level. In blue is the dimer, and it also inhibits about 50% of that level at 1 micromolar compound concentration. And you can see the dimer is more selective than the monomer. One of the things that we wanted to do, as I mentioned earlier, is that this compound is facilitating inhibition of processing, but what we were interested in is figuring out how to trigger the degradation of this RNA. And one ribonuclease that was well studied by Robert Silverman and colleagues is RNase L. And so RNase L is present in all cells as an inactive monomer, and it's activated upon a viral infection. And the way that it's activated is upon viral infection in a cell, cells will make this 2', 5' poly-A adenylate that'll bind to an active RNA cell, dimerize it, and make it active to facilitate targeted degradation. Okay, so the question we had is, could we co-opt this system to utilize 2', 5'A or some other molecule that could facilitate active dimerization to not cleave all viral RNAs, but to cleave RNAs that are brought into close proximity by binding a small molecule? And that's where we utilize this approach called ribonuclease targeting chimera, where basically we would take the microRNA 21 binder, append it with a small molecule, not this 2', 5' oligoadenylate, that would bind to inactive RNase L, and then that would facilitate this ternary complex formation to have local, not global activation of RNase L to cleave a target, and then it would allow regeneration of this ribotag. I'm not going to go into a lot of the chemistry here, but Silverman, in addition to studying the pathway, he also identified ligands like this. We did some medicinal chemistry on it that would bind to and activate RNase L. And all of this is in this PNAS paper. And so basically what we can do is we can assemble through this O-methyl these chimeras where we link this green molecule onto this RNA binding molecule. And then we had an ablated, inactive, it's too strong a word, so less active RNA cell recruiter that's shown here in orange, where we're attaching not at this O-methyl, but at this other O-methyl position. And then we make a series of active and inactive compounds. Okay, so we throw this into triple negative breast cancer cell line. And so in gray is the dimer, in orange is the control degrader that has this ablated nuclease recruiter, and what's in green is the active RNA cell degrader. And so what you can see is if we look at here, the levels of mature miR21, all of them will inhibit it because they can inhibit the processing of miR21 by binding to the dicer site. If, however, you look at the precursor to miR21 by RT-qPCR, you can see that both the dimer and the ablated ribotag inhibit, or they increase the levels of pre-miR21, which is supportive of it inhibiting processing. In contrast, the ribotag that's shown in green, the active ribotag, will inhibit the levels or diminish the levels of miR21, which is supportive of it facilitating targeted degradation. And we did a lot of controls to support this mode of action. Those controls include using an siRNA to ablate RNA cell from these cells will inhibit degradation. We can use an antibody that's directed against RNA cell. When we throw this compound into cell and do a pull down and do qPCR of what's pulled down, we'll pull down pre-microRNA 21, et cetera. There's a lot of controls that were done in this paper to support the assertion that we're facilitating targeted RNA degradation. One of the questions that we've had, and I just put the slide in here, is how selective are these molecules? And so this is just a quick selectivity. I'm not going to go into what a Gini coefficient is, but suffice it to say a number closer to 1 means more selective. So here's kinase inhibition. This is a Pfizer kinase compound that, if you go across the kinase tree, is more selective than staurosporin. It has a Gini coefficient of 0.91. If we look at monomer, dimer, and the dimer degrader, we can see that as we go from monomer to dimer, and here is the profiling of all microRNAs in TNBC cells, you can see that selectivity goes up as you go from monomer to dimer. Interestingly, selectivity further goes up as you go to a dimer degrader. And that's probably a consequence in that in order to get similar inhibition as observed for the monomer and the dimer and monomer require 1 and 10 micromolar respectively. The dimer degrader is 50 nanomolar, so it's 20 times more potent than the dimer is. And that's because these things turn over, as you guys are more aware than I am. They're substoichiometric. One of the things that we did in collaboration with Alex at Abikian's lab is we ran full proteomics to understand what was the consequence of these molecules on microRNA 21 mediated pathways, knowing that it's not really known. These are non-coding RNAs. And so this chemical probe can allow us to understand what are the effects broadly on the proteome by silencing microRNA 21 in a cell line. And so what you can see is that a lot of proliferative proteins are diminished in their abundance, and some anti-proliferative proteins are upregulated. If you'll recall, PDCD4 is a direct target of microRNA 21, and that's massively upregulated. And so one of the things that we can do is calculate what's called a context score. And that context score is basically, we can go, it's Chris Burge has written software, Target Scan, with Dave Bartel, where you can go in and look at all messenger RNAs and identify which messenger RNAs have a miR21 binding site in their 3' UTR. So what we can do is we can collect all of those messenger RNAs, and then we can annotate and ask a question and go, what is the effect of the protein abundance of those messenger RNAs that have a miR21 target site versus ones that don't? And so if they have a miR21 target site, they'll be generally upregulated, which is what we would expect because miR21 is a translational repressor. If they don't have that site, they're generally not affected. And so this is a tool that we've been able to deploy across the proteome to study what is the effect of silencing these non-coding RNAs, and it's giving us some insights into what their biological function is. There's a lot of questions that we can answer with these chemical probes. Last but not least is, I think this is one of the beauties of Scripps. I have a PhD in physical chemistry of all things. And so, being able to come and work in a, research institute like you guys are working in too, I don't imagine you would have any larger barriers than I would have, is we can take these molecules and ask questions in animals. And so what we were able to do is we can inject this dimer degrader at 10 mg per kg IP, and you can load in MDA-MB231 cells to a mouse tail vein. Now, I realize I'm talking to cancer specialists, and I'm by no means a cancer specialist. These are just the studies that we can do. We do other ones too, but we just wanted to see if this was active in an animal. And so here's vehicle. These are lung metastases of triple negative breast cancer cells that if you inject it in a tail vein, after a certain amount of time, you'll get these metastases in the lung. And then here's if we deliver the dimer degrader at 10 mg per kg IP, you can see diminishment in the number of lung nodules that are in this mouse model. And we can do HE staining. It's quite dramatic. These are just the bulk number of nodules, but the size and the number are both diminished. And this degrader, we were worried that we would overactivate RNA cell within the system and the mice would get very sick. It turns out that these compounds are totally tolerated within the mouse model. And furthermore, it appears that we're only stimulating a local RNA cell. So if we do all the markers of global RNA cell expression, we don't generally see it within doses that are efficacious. But if we do massively overload it, we do see those markers getting upregulated. But we're not massively overloading it to the point where we're having mice have issues when we're overloading it. For example, testing at the Cmax concentration. When we look at HE staining, we'll see normalized HE staining intensity is also diminished. And then the other thing that we've done within the mouse lung is we've taken histology and then we measured what are the levels of miR21. So we've done fluorescence in situ hybridization, and the dimer degrader is diminishing the abundance of miR21, mature miR21. We also looked and made a FISH probe for the precursor to miR21, and that is also diminished, right? About 60% in this mouse lung. And then we've also measured PDCD4. Remember, PDCD4 is a protein that's translationally repressed by miR21. And so we would expect that that would be upregulated. It's abundant, it shouldn't be upregulated. Its expression would be increased with this dimer degrader. And indeed, that appears to be the case in the animal. And so what I would say is that we have a mechanistically defined molecule that we have data to support that it's facilitating targeted degradation in cells all the way to an animal model where it's both the level of the RNA target and which are diminished, and the levels of the downstream proteins that are silenced by that RNA target are enhanced. We have been able to demonstrate degraders have broad function in diverse tissues in mice, and that suggests it's broadly applicable. We've been able to get these monomeric degraders that work in a brain. We've been able, as I showed you here, to get these direct oxidative cleavage of RNA to work in a muscle. We've been able to get these ribonuclease targeting chimeras to work both in a brain, in a kidney, in a lung. So we think this is important. One of the worries that I had with this, which I think you guys have thought a lot more about this, is actually the worries could be a benefit. is that in order for these things to work, you really have to have both your RNA target be expressed and the ribonuclease be expressed in the same tissue. And if it's a microRNA, it has to be the messenger RNA that is silenced by the microRNA to affect a function. And so, this multifactorial expression of these targets, I thought was going to be a nightmare in terms of getting these things to work. Now, I'm sure that is going to be the case. Working for us is a lower barrier than making a medicine, obviously. It's getting a chemical probe. And so the question becomes, what are the limitations of this? It's not going to be everything works and gives function. And so I think, being able to show that it works in diverse tissues in mice, I think might support that this could, be applicable to a bunch of different diseases to get chemical probes. Okay, so I'm going to conclude there. I don't know, doing science with people with many perspectives, I think is pretty fun. I grew up in a large family. As Brianna and I were talking, we both did. It's, when you grow up in a large family, you don't really have a diversity of opinion. And if there is, your dad will give you what the non-diverse opinion has to be, right? And so I think one of the things in science that's been really amazing, I mean, you have people I never thought I would not live in Baltimore my whole life. And I have people from Asia, from Europe, from exotic places like California in my lab. And so that's really amazing. And all of them bring unique perspectives that make science better. That's really, I think, one of the most amazing things in doing a scientist that I don't think you realize until you go out to the real world and talk to your siblings and know what they do on a day-to-day basis. I think sequence-based design within Forna can be used to inform bioactive small molecules targeting RNA broadly. What I wanted to highlight here to you guys that don't think about this all the time is there's many ways to degrade an RNA by using small molecules. And I think we're just at the tip of the iceberg. You can get allele selective molecular recognition in animal models by using these structure binding ligands that you can profile by RNA-seq. That is going to be really important. Huntington disease clinical trial came out last week, and the ASOs are not targeting the repeat because it's very hard to get things to be selective at the repeat. I don't know what this means, but maybe an alternative is to have structure binding ligands that directly get at the repeat. And then ribonuclease targeting chimeras can cleave RNA targets catalytically and substoichiometrically in cells and animals. And I think there's a lot to do. I think that's I got into this. I tell people, being from a large family, you're very competitive in many ways. And I think one of the things in science that I wanted to do is sort of have a white space to work in. And I think that there's just so much to do in the RNA space that, my whole career would be devoted to this, and I don't think we'd be scratching the surface of things to do. So thank you very much. I want to thank my students again. I started thanking them at the beginning. So thank you guys for listening to me, and I'm happy to plow through these questions.